Hi everyone, I'm Saket. I'm probably going to be one of the outliers in this workshop because I'm a chemical engineer turned computational biologist. I'm neither a clinician, nor a doctor, nor a oncologist in that fashion. But I'll give you some vignettes of things that I've done, which hopefully you'll find useful in your research. So one of the goals that biology tries to understand is it tries to understand what shapes a particular tissue, what shapes a particular organ, what is the molecular composition of a particular tissue, right? So I'm interested in brain in particular. And one of the focus that we want to essentially understand is how do we understand the phenotypic diversity that arises because of molecular diversity. And we all know, we are all familiar with, there are ways to capture molecules now, right? So how do we actually capture molecules? We mostly my lab focuses mostly on understanding RNA, just because the signal to noise ratio is generally higher when you capture the RNA. And if I'm interested in brains, brain is made up of different cell types, neurons, oligodendrocytes, astrocytes, very broadly. The goal here is, can I profile the RNA molecules from these cells, right? So none of it is new. We have been doing this for multiple years now, almost 1.5 decades now. So this technology which I'm going to call bulk RNA-seq, and you'll see why it's called bulk. This technology came out around at the end of 2008. The idea is you want to capture the molecules, the RNA molecules of the tissue. You start from a tissue, you also isolate the RNAs. This is easy to do because they have poly A tails. You can generate cDNA, fragment them, size select them, and put them on a sequencer. And when you get this sequencing readout, what it tells you is what is the molecular makeup of each tissue. This could be one condition, it could be a tumor, it could be a cancer, it could be a normal sample. So if I were to sort of visualize this, if I were to visualize in terms of what information RNA is really capturing, I have a schematic to sort of help you visualize. So again, I'm visualizing the brain. Brain is made up of different cell types, which here are visualized as different colored Lego pieces. When I do bulk RNA-seq, what happens is while I'm capturing the average molecular makeup of that particular tissue, there are two levels of information that go missing out. One, the spatial location of where exactly that molecule came from, that's totally missing. The second is which cell type it actually originated from, right? So a cell type is supposed to have molecules which are doing very similar function in that sense. What you get out of this bulk, and that's why I call it bulk RNA-seq, is you get an average molecular makeup of the brain. So this paper came out in 2008, is extensively used everywhere, including in cancer literature. Just the following year, 2009, people started to think about how do we solve that one problem of not being able to retain the cell type specificity. So they developed this technique which is called single-cell RNA-seq, which I'm going to abbreviate as scRNA-seq, where you do very similar steps, and I'll show you what these steps are. But what it helps you to retain is the average molecular makeup of individual cell types. So you still lose out the spatial locations, but you retain the specificity of which cell it is coming from. Any molecular workflow of single cell, there are multiple technologies now. They use these broadly 4 steps. The first step is you dissociate the tissue. You physically separate them into cells, right? So I told you that brain is made up of broadly 3 different cell types: neurons, oligodendrocytes, and astrocytes. You perform dissociation, you perform and then you physically separate these cells. Then you perform a lysis within that particularly separated cell, right? So if I perform lysis, the mRNA molecules will come out. The moment they come out, they are actually also tagged by a unique molecule, which is called the barcode, which is what you see in colors here. Once you have these barcodes, you can then just do a RT and then throw them on a sequencer. Since the fact that each molecule that's coming from each individual separation is unique, you can tell which molecule came from which cell. And that's how you retain the cell type specificity of this information. So I just want to give you a quick vignette of what are the challenges when you start to look at this kind of data. So one way to sort of visualize how this field has been growing over the past decade is to look at, is to contrast it with the number of cells that we have in human body, right? So we have around 36 trillion cells in the human body. One way to compare the scale at which anything is growing is to contrast it with what's called Moore's law, which says that every 2 years, the number of transistors will double up on your microprocessor chips that you're using right now in your laptops. So this grows by Moore's law. On the right side, what I'm now comparing this is the total number of cells that have been profiled using this technology in unique data sets over the last decade or so. And what you'll see is the slope is much higher. So the data is growing at tremendous scale. As the data keeps on growing, as a computational biologist, I have to think about what are the methods that I can sort of leverage this data in the right fashion, right? So for every single data set that gets put out in public, people have deposited, for every 4 data sets that turn out to be, that are deposited on public repositories, we have 3 methods coming out just to analyze that data. So why am I highlighting that particular challenge is because when you have to model this single-cell RNA-seq data, you have to really think about from first principles, how do you actually normalize data? So what I'm highlighting you here is, so I developed methods, I developed statistical methods for analyzing genomics data in particular. This is a method that I developed towards the end of my postdoc, where we try to sort of look at how do we normalize single-cell data. So this tool is called SC-Transform. We developed this because I'm going to show you some biology which was useful for, this tool was useful for doing that biology, and it has been widely adopted by the community, which is very reassuring. Okay, so what is the biology that I'm interested in? So I'm interested in, if you have information from one source, how do you actually leverage that source to tell you the information that is unknown? And I'll give you a vignette of 2 different species, and then you can think about multiple examples. So if you have patients coming from different cohorts, how do you actually analyze that data? That's kind of the vignette, this particular vignette is inspired from. So I call this talk comparative, and the comparative sense here came from my love for evolution and my love for understanding how, again, phenotypic diversity arises. So we have seen tremendous developments in the genome after the human genome came out. And one of the things we try to do normally is we contrast what humans have in particular, which is not there in other species, right? So I'm showing you here an example of a gene that's ultra conserved. There are certain sequences that are not conserved, and that's probably the reason certain species behave in the way they do. So the goal here was, if I profile single cell data from 2 different species, right? So this is ferret and pig. What you're seeing here, each individual white dot here represents 1 single cell. I have no idea right now of what these cell types are doing. I know in a lower dimensional, I see some clusters, so there is some information, but I have absolutely no idea what these cell types are. So I'm neither a biologist, I'm neither a neuroscientist, so I have no idea actually what is going on, right? But people who came before me have actually done tremendous amount of work on motor cortex. So what I'm showing you here is the profile of motor cortex profile from ferret and pig. Particularly, there has been a tremendous amount of work that has happened understanding the motor cortex in mouse. I'm showing you here an example of single-cell data profiled from the mouse motor cortex by the Allen Institute. And you can see this is beautiful colors, right? So what these colors represent is each dot, again, is 1 cell. And these different labels are the different cell types that mouse has. So what I did is I said that, okay, if I have this information available, I can actually leverage this information computationally to tell me what happens in ferret and pig. So what I did is just that the way you do human genome alignment, I'm actually doing transcriptome alignment at the single cell level to tell you what cell types are present in ferret and pig. So I use this as a reference. You can visualize this as doing genome alignments, but doing this at the single cell level, at the transcriptome level. And what I'm showing you here is that this, I'm skipping out all the mathematical details of this method, but it works beautifully. What I'm showing you here is that once you do that kind of projection, things sort of profile in the same, they follow in the same trajectory, which means that you're able to annotate using mouse data, ferret, and pig data, right? And you can sort of imagine, you can do this across different donors, you can do this across different conditions. So you have a normal versus a cancer patient, you could do something similar if you were to profile single cell data from cancer patients. So very quickly, one of the results I wanted to highlight is why am I doing this, right? So one of the reasons we are interested in is using this technique, I was able to identify a new novel cell type that was previously claimed to be primate specific. So it was present in humans and macaques, but not in ferret and pig. We show that this cell type, which is TAC3 interneurons, these are actually only present in pig, but they're not present in mouse and ferret, and they're not primate specific as what was claimed previously. So very quickly, I talked only about single-cell RNA. It doesn't tell you everything that's going actually on in the cell. We now have the ability to also profile what is going on in terms of at the DNA level, or particularly at the open chromatin regions, which is where the transcription factor binds and which is how gene gets regulated primarily. So this technique is called single-cell attack-seq, which basically, in brief, tells you which regions in single cells have open chromatin, right? So this is where transcription factors will bind. So what the current focus of the lab right now is so there was a lot of talk about heterogeneity. So first of all, I should have started by just thanking the amount of information that I've been able to capture in the last 2 hours, which is there's a lot of talk of heterogeneity, there's a lot of talk of DPI and ML. So one of the things that the lab right now is focusing on is understanding how sequence changes determine what changes at the expression level in individual cell types. And the way we are doing this is by leveraging information across species. So very briefly, what we do is we take sequences from multiple organisms which are coming from open chromatin data, and then look at train deep neural networks to tell us what particular single nucleotide change will have impact on the expression level at individual cell types. Very quickly, so I'm a faculty member at the Center of Digital Health, and I'm showing you data from mouse and ferret and pig, and like what exactly am I doing, right? So I had the same question in my head, right? So I came back to do some translational work, and this has been possible through the collaboration that we have with Tata Memorial Hospital in particular. So one of the projects, the recent grants that we wrote with Dr. Seema Gulia and Dr. Sharmila was to understand at individual cell type level, what are the molecular changes that happen across different grades of neoplasia, and what impact the actual impact of the expression of virus is doing at what impact the particular virus protein levels is having on the in terms of what we see at the host level. So what is the host pathogen interaction at single cell level? So very quickly, this is my last slide. When I'm showing you that we are able to sort of do serotype, I'm just profiling single-cell RNA-seq data. This is, by the way, a public data set. We're just profiling single-cell RNA-seq data, but with just that information, we're able to find out what that particular serotype of the HPV infection is, how much protein is present in individual cells. So this is what is shown at the bottom. And with that information, the goal here is to understand the impact of the expression pattern of this particular cell, of the viral protein on individual expression of the gene coming from the host. Okay, so very quickly, what I've been able to tell you is, hopefully, is that if you profile data from multiple single cells, that's one way to capture the heterogeneity that exists in any particular tissue. And you can use single-cell multiomics to provide you mechanistic insights into what causes a particular disease. So I just opened my lab 3 months ago, so I'll use this opportunity to advertise it as well. So we are funded, we are hiring at all levels. So if any of this interests you, please reach out. I'm happy to take any questions. Thank you.
And only one question, if anyone is asking.
So you said SNVs which are evolving based on the multi-organism genomics that you want to correlate with the cancer SNVs. But the SNPs which are evolving through multiple organisms, they are characteristically very much different from the somatic mutation profile. So how are you factoring for
So
that?
great question. So I think I confused probably because I was going very fast. But the thing that we are actually leveraging is we are leveraging whether if something is conserved across different species, it likely is functional, and that's why it's conserved. So it's carrying out very specific function. What we are trying to do with these deep learning neural network models is to understand what is the sequence regulatory code that is conserved, because that is what is, so given a particular sequence, this is the expression. So this is the sequence coming from the cis regulatory element level. What is the corresponding impact that it has on the expression? And we do this across different organisms. This is sort of learning what sequence architectures exist across different organisms which has stabilized the gene expression. So we're trying to essentially focus on what is conserved rather than what is novel. Then once you do this training, you can come back and say that, okay, if this is the mutation, what would the likely impact of it have on the expression level?
Are you also looking to the epigenetic effect on these changes?
Great question. So Yeah, so we are looking at epigenetics, but from a very tangential point of view. So I'm actually interested in looking at single cells, and the single cell epigenomic part. You'll see multiple papers, but I think I can tell you like none of them actually work. So the problem is doing this, any sort of methylation data at scale is hard. It just doesn't work. There are technologies to profile individual transcription factors. Those actually are robust. So we do plan to use that and eventually sort of do this in our lab. But so far the focus has been on tangential. So we just look at attack-seq, which is just showing you the open chromatin region. So you know that some transcription factor binds there and there is some change there, but it doesn't tell you more than that. But so far, as a proxy level, that's been something which has worked out beautifully. But that's not like panacea for everything that we want to do.
Thank you, Dr. Saket.
Great.